ONI is a company that is focused on developing technology and enabling researchers to solve the most difficult problems of our time using super resolution microscopy. Our mission is to simplify super resolution, and we're doing that through a set of products that work end-to-end from how a researcher begins their experiment with sample prep all the way through to analysis. ONI exists to transform the way that discovery is done, whether it's therapeutic discovery or basic biological discovery. We believe the fastest path to discovery is often seen, and our part of that is enabling researchers to see and to quantify at the molecular level.
Super resolution microscopy is a way for us to see things that are much smaller than we'd typically be able to see with a light microscope. Light microscopes are typically limited to about 200 nanometers of resolution. And what that means is if objects are closer than 200 nanometers apart, we can't actually distinguish them. But what super resolution microscopy allows us to do is we can use some computational tricks, some chemical tricks, and then some computational power to bypass that 200 nanometer diffraction limit, and then we can actually see things that are up to 20 nanometers apart. So really, really small details inside of things like cells, tissues, or we can look at things like nanoparticles, which are typically under 200 nanometers to begin with. So some of the ways this has made an impact is when you're looking at things like macromolecular complexes within a cell, where previously you might have basically seen what accounts to one giant blob. Now, when you look with super resolution, you can see all of these different subunits that are actually making up this complex as a whole. And what this also allows researchers to do is they can individually knock out different components of that and see how it affects an organism or a cell on a whole. So this level of detail allows you to not only investigate where things are spatially, but also allows you to get more information about what the actual function of something like a subunit within a complex does.
ONI has a broad range of customers, from pharmaceutical companies to biotech and academic research labs. Our super resolution platform supports a wide range of different scientific applications within the life sciences and medicine. These include applications such as cancer research, extracellular vesicle research, virus research, neuroscience, just to name a few. With the ability to support so many different scientific fields, it shows how powerful super resolution microscopy can be in supporting our customers and helping them to make good scientific conclusions within their respective fields. Accessibility is a huge pain point for our customers because traditionally these super resolution microscopes can be quite hard to use, they're not intuitive, and they're often not accessible unless you go to a microscopy facility. So we address this challenge by designing a desktop-compatible system which has been designed to simplify the super resolution workflow. Our super resolution microscope can operate on any lab bench, removing the need for specialized infrastructures and optical tables, and we provide intuitive softwares and automated solutions that allow our customers to get to their results much quicker and efficiently analyze these complex biological samples.
ONI was founded in Oxford, and it had a particular point of view even then that is consistent to what we're doing today, which is that super resolution is important and very valuable, but it's also really difficult. And so the company has always been about simplifying super resolution microscopy, making it more accessible and making it more repeatable. ONI offers the simplest super resolution microscope in the market. It fits on a desktop and it resolves down to individual molecules. Also, ONI offers application-specific kits. Our CODI software, so CODI stands for Collaborative Discovery, it enables the acquisition and the analysis of super resolution data. The newest addition to ONI's product portfolio is AploFlow. It's both sample preparation and fluidics, and it enables researchers to complete their super resolution experiments with significantly less hands-on time, and it improves the repeatability of results.
The way that super resolution can really change how people think about their therapeutics and their research is it's really going to add that extra level of detail. Imagine suddenly you woke up one day and you could see 10 times more detail. There's so many new things you'd be able to notice. You might notice that maybe some flowers look slightly different than other flowers. That bee has a certain stripe that that other kind of bee didn't have. And once you can see that level of detail, you can start asking new questions. Okay, why does it look like that? Why is this molecule in this part of the cell? So once you can see more details, you're able to ask more questions and get even better answers.
We are equipping our customers with user-friendly imaging technologies, automation, and streamlined workflows, which allows them to get to their results quicker and more efficiently. Enhancing the reproducibility and reliability of our products enables our customers to confidently get consistently reliable results, which will help them make advancements in their scientific conclusions.
Super resolution enables a much more direct understanding of mechanisms of action. So whether that's associated with antibody drug conjugates or exosomes or LMPs, what super resolution enables is for people to very directly study the mechanism of action. For the next year, we're going to broaden the set of things that customers can analyze with our workflows. And think of it as the journey of other techniques that are more mature: DNA sequencing, PCR, other techniques where essentially it's push button, it's end to end. And that's where we're going with super resolution. The innovations over the last year, and those that we're going to be coming out with soon, mean researchers focus less on the method, less on how to get the result, and they focus more on what to do with the results.